Hey everyone, so this is the second lecture for PSY4050 Quantitative Research Methods. So this is the week 3 lecture, and I was just running through the lecture that I've written to see if I could work out the timings. I thought what I might do whilst I was doing that would be make a recording of it, which might be useful if people watch it another time. Okay, so this week, what we're talking about are descriptive statistics, distributions, and statistical significance. Those are the things we're going to cover. The aims are to discuss the importance of inspecting and visualising data, so getting familiar with your data before you analyse it. We're going to review some descriptive statistics, introduce and discuss data distributions, and also talk about null hypothesis significance testing. So quite a lot of material really to cover in an hour. Okay, so at the end of this lecture you should be able to calculate measures of central tendency and dispersion, interpret graphical representations of data, understand probability distributions and the normal distribution, and demonstrate an understanding of statistical hypothesis testing, specifically null hypothesis statistical testing, or null hypothesis statistical tests. Okay, so first of all, what is statistics? Statistics is where we use data to try and answer interesting questions. And data is information we collect in relation to a particular question, subject, or issue that we're interested in doing research on, and there's variation within that information. There will be variation in traits in any natural population, and entities will vary in whatever it is that you're trying to measure. In some disciplines, that is more the case than others. So social science disciplines like psychology tend to have very noisy or messy data. And the role of statistics when we collect data on a particular question is to allow us to present and analyse the data to try and answer some sort of research question. Okay. So types of statistics. Hopefully a lot of this is revision or review for most people on this module. There are two main types really, or we could split it into two. They're descriptive statistics, which we use to summarize data, and this means that we can use large quantities of numbers to try and capture patterns with a few simple measures. Okay, and contrast to that, we've also got inferential statistics, and this is where we use probabilities to make inferences about population on the basis of samples of data. Okay, so when we use inferential statistics, we're usually trying to test hypotheses, make predictions, or assess relationships and associations in some way. We're going to discuss both of these on this module, but today essentially we're going to focus mostly on a little bit of descriptive statistics and then some conceptual ideas. But before we do that, before we start conducting some sort of analysis when we've got data, a good place to start is by getting familiar with the data that you have. Okay, so a basic data science workflow for someone using data to answer behavioral or psychological question might look like this slide, essentially. So we want to get familiar with the data set that we're working with, what the variables are, what scales we used to measure those variables. And the first step to that is we want to inspect our data. Is it tidy data? Are variables are collected together in columns in our data set? Is it set up like a panel data, like a spreadsheet? And are our observations, are they allocated to rows, so it's easy for us to make sense of the data sheet that we've got, the data set that we've got? And we should look for missing values and things like that, other issues like that. For example, anomalous data scores out of range. Okay. And once we've done that, we probably want to clean, or what in data science they refer to as munging. We want to munge the data and when we're cleaning the data, we want to consider the possible impact of any problems that we've highlighted when we've inspected the data and make a decision about what to do about them. Maybe we want to delete entire variables because there's so much missing data. Maybe we just need to delete individual cases for some reason or another. Okay, and many software packages that we use for statistical analysis can handle missing values quite well. If you have a lot or lots of missing values on one variable, it may result in erroneous conclusions for certain analyses, because then you may end up comparing sort of different subsets across variables, and that might not be a useful thing to do. Okay, so the next step, we might want to visualize our data and the distribution of scores that we've got. This will help us understand whether our data deviates significantly from a normal distribution. I'm going to say more about that in forthcoming slides in this presentation. And whether we may need to be cautious when conducting parametric statistical tests if it does deviate significantly from the assumptions that the data is normally distributed. Okay. And then after that, we can go on to obtain some descriptive statistics and then run some inferential statistical tests, such as things to test our research question or hypotheses. Okay, so graphical representations of data. Okay, so the next few slides, I'm going to run through a few slides showing how visualizing data is helpful for us when we're trying to understand our data. On this slide, there are a couple of types of graphical representation of data listed: histograms, stem and leaf plots, and box plots. I've grayed out stem and leaf plots because I think the other two are much more useful. So stem and leaf plots, I've never really seen them used in a particularly useful way, or never seen them in a journal article or research article of any type. So I'm not really going to cover them in depth. They're largely like if you got a histogram and turned it on its side. They're like that, only they contain a little bit more information. But I'll focus more on the other two, histograms and box plots. They can be quite useful, because they allow an examination of the shape of distribution, and they have strength. Each type has different strengths and weaknesses. Some things that are worth commenting on, though, are the fact that data can be the shape of distributions of data can be skewed in some way. So we can find we've got negatively skewed distributions where the median is larger than the mean, and the tail points towards the negative or low numbers. Okay, so the skew relates to where the long tail of the distribution is. We'll see what this means in a second with some pictures. Alternatively, they could be positively skewed distributions. In this case, the mean's larger than the median because the mean's been pulled up by kind of extreme scores in a long tail at one side of the distribution. And the tail in that case points towards higher numbers. Okay, so here's a histogram. This is, I think, it's data from some sort of arithmetic test, and it's on the x-axis, the horizontal axis, you've got scores on the test, I think out of 20, and then on the y-axis, the vertical axis, you've got the frequency, the number of people who got that score. So we've got a total sample here of 174, n equals 174. With a mean score on the test of 9.1, a standard deviation of 2.43. Okay, and this histogram, you can see it's quite useful. It gives us an idea of the shape of the distribution of scores that we've got, and we can see from looking at it there that about 50 people got a score maybe of 10 out of 20, something like that. Okay, so another quite useful graphical representation is the box plot. Probably all seen these before, sometimes referred to as a box and whisker plot. Okay, so the square box in the middle is that contains the middle 50% of scores, so the middle 50% of our distribution, and the two long lines going out either side contain the sort of lower and upper 25% of scores, excluding extreme values or outliers as such. And as you can see, now the axes are slightly different here in comparison to a histogram. You've got arithmetic shown on the x-axis, and just the number of individuals, 174, and then on the y-axis you've got the actual scores. Okay, often when you get these, you get a line through the middle of that box in the center, which shows you where the mean is. On this one in particular, you can see there's about above the top whisker, you've got a number 49, so that will be participant 49, and they're an outlier because they've got 20 out of 20. So this is the same data as we saw previously in the histogram, and you can see, although it's the same data, you can see there are different kind of benefits to different types of distributions. Sometimes it helps to have both. So we can see from this that a lot of the scores, 50% of the scores, are clustered around between 8 and 10. And we've got kind of one extreme value. It wasn't as easy to see that on the histogram, and we can also see that the longer tail, which was the positive skew we saw on that histogram, is to the higher value. So if you look at that whisker for the top 25% of scores that comes out the top of the box, that is longer than the one at the bottom. Okay, right. So positive skew. Here's a graphical, just to sort of give you a real clear graphical representation of it. So a distribution like this, we describe it as positively skewed. It's got a long tail going to the higher numbers. If you find that's intuitively a bit confusing, I used to think positive skew would mean most of the numbers are up towards the positive end, but it doesn't. It relates to the tail. So it means a lot of the scores are at low numbers, but there's kind of some small frequency of values going up to the higher numbers. Okay, as you can see, there's like 1 or 2 people that have got scores of like 60 and 40, things like that. Okay. The opposite of that is the negative skew. So here we've got the tail going, this long thin tail going down towards the lower numbers. Okay, I found this online. It's quite a useful image, I think, and it shows you kind of how skew affects the mean, median, and mode. And if we have a symmetrical distribution, like a normal distribution, the one in the middle, we're going to talk more about that in a sec. If we have that, then the mean, median, mode are generally identical, and the distribution is symmetrical about the mean. If we've got positive skew, because we've got these extreme values, it pulls the mean up, so the mean is higher than the median in this case. So hopefully you guys understand that the mean is not a robust measure of center, because it is susceptible to extreme scores, because every score contributes to the mean, where it's not the case with the median. We will cover those things again shortly, or we'll revise those things in a couple of slides' time. The opposite you can see there with negative skew, the mean is lower than the median. Okay. I found this as well in a textbook, Open Interest Stats. I thought it was like a useful diagram, and it shows you kind of a box plot next to a histogram. You can see the kind of strengths and weaknesses of why it might be useful to use both when you're visualizing, getting familiar with your data. So strength of the histogram in that top image there, as you can see, there's like this bimodal distribution going on. You've got 2 peaks there, one around 5 and one around 12, 13. You can't see that clearly in the box plot, but the box plot does give us a better idea with the line there through the middle of where the mean sits. It's roughly around 10, and it also gives us an idea that we've got 3 or 4 outliers in that data set as well. Okay, so let's move on to descriptive statistics. So 2 important concepts in statistics are center and spread. So center, particularly measures of what are known as measures of central tendency, are an attempt to use like a single measure to describe the patterns present in a data set. Spread looks at how much variability is present between the observations and data points in a given data set. And the concept of variability comes up a lot in statistics. A lot of statistical analyses involves trying to quantify and measure variability. Okay, so just a quick review, bit of revision hopefully here. So measures of central tendency, three most common ones are the mode, which is the most common score. If you've got a data set, whichever score occurs the most, that would be the modal score. The median, which is the middle score. So if you've got a data set and you rank order your scores in that data set from lowest to highest, then the median would be the middle value. So if you had an odd number of data points, so say you had 21 data points, and you rank order them from the lowest to the highest, then the median would be the 11th, the one that was ranked 11th in your data set. So it had 10 data points below it, 10 data points above it. If you've got an even number of data points, such as 20, then the median in that case is going to be the average of your middle two data points. So that would be the average of points, the 10th and 11th data point as such. The mean, much more commonly used measure of central tendency, is the average, essentially the arithmetic average. And you can see there a couple of mathematical formulae for how you calculate the mean. The top one there is the population mean, and the bottom one is the mean for a sample. Okay, so hopefully you recall from last week, bit of notation revision here. So Greek characters are often used to represent population parameters. Latin characters are often used to represent sample statistics, and capitalized letters are often used to represent population parameters, and lowercase letters are often used to represent sample statistics. Okay, so the top one, we've got the Greek letter mu is equal to, and then we've got this the Greek, capital Greek sigma, character sigma, which, as I told you last week, that means, if you see that, it basically means sum everything that comes after it. So we've got capital Greek character sigma, and then x divided by capital N there for this population mean. And x is a score, and if we had, basically what it's telling us to do is sum up all the scores in our data set, then divide by how many scores we have. I think most people understand that. That's how you work out an average. The formula is the same. It's the same calculation, but the formulas are obviously different when you're specifying whether it's a population mean or a sample mean. Most of the time, we're going to be working with a sample mean because we won't know what mu is or capital N, the population mean and total. Okay, so spread, measures of dispersion or variability. There's a few different ones. There is the range, which is the difference between the largest and smallest score. It's not very informative, but it can be useful to report it when you write up results, because it can give you an idea of kind of how skewed data is. So if the lowest score in your data set was 2 and the highest score was 18, the range would simply be the highest minus the lowest. So it'd be 18 minus 2. The range would be 16 points between the lowest and highest score. And if you couple that together with knowing what the mean is, that kind of give you an idea of how skewed your data is. So people often report range. The interquartile range, that's the range of the middle 50% of scores. On those box plots, it was the box bit in the middle, essentially. The interquartile range can be quite useful because it's a robust measure, because you essentially exclude the top and bottom 25% scores when you work that out. Okay. C, a kind of an interesting one that you don't hear talked about much is what's called the mean absolute deviation, or the MAD, the MAD. So you can see the formula for it there on the right slide. So the formula is you basically work out the mean for your data set, and then you subtract every score from the mean and divide by how many data points you have in your sample. But when the way the mean is constructed, some of your data points will be above the mean, some will be low, so some will be negative numbers, some will be positive numbers, and those things will cancel out. So to deal with that, you take the absolute value, and that's what these two, you can see these two vertical bars after the sigma sign there in the numerator, the top part of that division for the MAD. And you've got a straight vertical bar, then you've got x minus x bar, which is the sample mean, and then another vertical bar. And those two vertical bars basically just mean take the absolute value. The absolute value being the distance from 0. So effectively you're ignoring negative scores when you do this. So to give you an example, if the score was 4 and the mean was 6, that would be 4 minus 6, which would be negative 2. The absolute value of negative 2 is just 2. If the score was 8 and the mean was 6, that would be 8 minus 2, which is just 2. The absolute value of which is still 2. So essentially, when you're working at absolute values, you're kind of ignoring negatives. You're just looking at how far from 0 it is. Okay. The mean absolute deviation is quite interesting, but it is rarely used. I think I've never really seen it used. A tendency is for people to use the variance, and there are several steps to calculating the variance. It's quite interesting. It's quite an interesting formula, I think. Firstly, you need to calculate the mean for the data, and then you take the difference between every data point and the mean. We're not taking absolute value in this case, so some of the deviations will be positive and some will be negative, as some points will be above and some below the mean. The third step is then to take each of these differences and then square it. Okay, this gets rid of negative values that would cancel with positive values. And it also increases the weighting of scores that are further from the mean. Of course, we're raising those distances to some exponent. We're squaring it. Okay, so the fourth step then is to take those squared differences and sum them all together to give a single value called the sum of squares. The sum of squares is then divided by the number of independent pieces of information in the data set, which is the number of data points minus 1 in the case of the sample variance. Okay, this is, so the number of independent pieces of information in the data set is known as the degrees of freedom. And I will illustrate this, I'll kind of go into more detail about that and illustrate what that means in the lab class in more detail. Okay, so those 5 steps give us the variance of the data set. But the sum of squares on its own is uninformative because the size of the sum of squares is related to how many data points there are in the data set. The more data points, the larger the sum of squares will be, hence the need to divide by n minus 1 for the sample variance. Okay, so the variance provides a measure of spread using square values, so it can be difficult to interpret in a meaningful way. So the variance actually gives us the average squared deviation from the mean, and that's different from the original sort of measures that we used to record that data. Okay, so that's not very helpful, but we can rescue the situation with the standard deviation. Okay, so the standard deviation, our last measure of spread that we're going to cover at the moment. So this issue with the variance not being in the same units as the original data is kind of resolved using another measure of spread called the standard deviation. And the standard deviation is just the average deviation around the mean for a given data set. It has the same units as the original data set, and the standard deviation is simply the square root of the variance. If you look at the formula that are on this particular slide, the top one's the population standard deviation, the bottom one is the sample standard deviation, and you can see there's a square root radical sign. Everything under that is exactly the same as the formula for variance. So we just take the square root of the variance. That gives us the standard deviation. So y divided by n minus 1 for sample variance and standard deviation. So in formulas for sample variance and standard deviation, we divide by n minus 1 rather than by n, which is used in population variance. And we do this because we don't have n independent pieces of information about variance. Okay, the first step was to calculate the mean for n independent pieces of information or data. The second was to calculate a variance using that mean. If n minus 1 deviations from the mean are calculated, it's kind of known what the final deviation must be, because they must all add to 0. It's just a function of the way the mean is calculated. So the last piece of information is determined, and we have n minus 1 independent pieces of information on variability about the mean. Okay, so if we divide by n, we would get an underestimation of the variance. As I said, what I'll do is I'll go through an example in the lab class, which kind of illustrates this. Right, let's move on. Okay, so a quick review of probability, and then we'll go talk about distributions. So hopefully this is just revision for most of you guys. The probability of an outcome is the proportion of times the outcome would occur if we observe this process lots of times, so an infinite number of times. And probability is defined as a proportion. It takes values between 0 and 1. So 0 would mean the event never happens, 1 would mean the event always happens, and values in between, the closer to 1 a probability is, the more likely it is to occur. The closer to 0, the less frequently it occurs. Okay, but it's often the case that probabilities are displayed as percentages. Okay, so some examples there. If the probability of event A is 0.5, then we could also say the probability of event A is 50%, or there's a 50% chance of event A occurring. So if that related to coin tossing or something like that, the probability of getting a tails is 0.5 or 50%. Okay, right, distributions. So the most basic distribution is the frequency distribution, where we list all values of our variable on the x-axis, so the x-axis being the horizontal axis, and the frequency or number of times they occur on the y-axis. From this, we could work out the probability of getting a certain score based on how many times it occurs relative to all observations. We can make this more useful and interesting, though, this type of distribution, by converting it to a probability distribution where, instead of recording the frequency of events on the y-axis, the vertical axis will record the probability. Okay, and here I've just put the histogram we looked at earlier in relation to arithmetic scores and frequency. That was a frequency distribution, essentially, for that particular variable. There are mathematical formulas for converting a frequency distribution to a probability distribution. If your data is discrete, like this one, so each score, values can only take, scores can only take on certain values, like 2 out of 20, 3 out of 20, 4 out of 20, etc. Then it's pretty trivial and straightforward to sort of convert this to a probability distribution using something called a probability mass function, or PMF. Okay, you don't need to do this. But if you have continuous scores, the function is difficult to work with, and it requires a lot of computation. If the scores that you were recording were continuous, then you would need to use a mathematical function called a probability density function, and they're notoriously difficult to work with. So why is this important? Well, the inferential statistical analyses we use rely on probability to allow us to draw conclusions about events and patterns in our data. So these statistical tests are based on or underpinned by probability models. And these are models that give us the probabilities for common distributions. So rather than having to try and work that out from a frequency distribution ourselves, we can look at certain probability models. Okay, so a probability model, as I said, underpins the statistical tests that we use, and they're specified by two specific parts. Firstly, a random variable, which is simply the variable we're measuring, and we're using numbers to quantify some trait or process. So it's a mapping of a random process to an outcome. So if we're looking at recording height of people, then whatever scale we use to measure their height, that would be our random variable. Height would be our random variable, and we would quantify their heights using numbers in some way, on some measurement, centimeters, inches, whatever it is. Okay. The second part to a probability model is a probability distribution, and that's a plot that describes an idealized frequency distribution for a variable, from which we can obtain the probability with which specific outcomes will occur. So we kind of list all values, and then list all the values on our measure, our variable, and the probability of occurrence. So we list all the values on the x-axis, the probability of occurrence goes on the y-axis. And the common statistical tests that we use work on the assumption that our data is similar to the probability model that the test is based on. So if our data isn't similar in distribution to the probability model that the statistical test is using, it can lead to erroneous results and conclusions. Okay, so there are lots of different models and probability distributions. One model that we're particularly interested in is the normal distribution. It's also called the Gaussian distribution. Okay, and a lot of the variables we might be interested in measuring are distributed in this way. The normal distribution has been central to the development of a lot of statistics, as a lot of the most common tests we use make use of the normal distribution as the underlying statistical model. These are what we often refer to as so-called parametric tests. So the normal distribution is bell-shaped. It's symmetrical about the mean. The mean, median, and mode have the same value. We saw that earlier in a symmetrical distribution. It's asymptotic, which means if you look at the tails as they head off in different directions, they never touch zero on the horizontal x-axis. They become asymptotic to the x-axis and go off towards negative and positive infinity, okay, without actually reaching zero. And the normal distribution is defined by two parameters. Those are the mean and standard deviation. So you'll notice from looking at this image that the normal distribution is a continuous probability model. Okay, that doesn't mean you can't apply it to discrete data. You can. Remember that this is an idealized probability distribution. But because it is continuous and can be specified with infinitely fine precision, outcomes take on values over a continuous range. Yeah. So we can find the probabilities of intervals, such as probability of a score being less than or greater than some value, or between two values, but there's no area over a single point. Okay, so the area under the curve here on this particular normal distribution would sum to 1, essentially. Hence the reason we're able to work out probabilities. So as I stated previously, working out probabilities for a continuous distribution using a probability density function is quite difficult. Fortunately, statisticians have worked out a standardized version that's much easier for us to work with, and that's called the standard normal distribution. Okay, so it's also called the Z distribution. So the standard normal distribution has a mean of 0 and a standard deviation of 1. And any normal, roughly normal distribution can be transformed into a standard normal distribution via the Z-score formula. It's important to note, though, that if your data isn't normally distributed, standardizing it in this way, it won't make it normal. it will just put the mean and standard deviation on standardized scales, on different scales. Okay, and there are well-established tables called Z tables that can be used to look up the probability that a score lies between any two values. Okay, so the Z score for individual observations is the number of standard deviations above or below the mean. This allows us to make inferences about the likelihood of events. Okay, so that's all quite theoretical. Let's kind of look at a Z score example. So we can work out Z scores for individual. We can work out the Z score for individual scores or individual data points. Okay, so for example, say that we know that a standardized reading test for 9-year-old children has a mean of 100 and a standard deviation of 5. So mu is 100, sigma is 5. So that's the population mean and standard deviation. So it's obviously been standardized extensively. Okay. If a particular child scores 90 on the test, do they need extra help with their reading? So there's a research question. So first thing to do is to convert their test score to a Z score. We do that using what's known as the Z formula. So that is Z is whatever their score is, X minus the mean divided by the standard deviation. So in this case, the child scores 90. We subtract the mean, which is 100, and divide by 5. What we get there is negative 10 divided by 5, which gives us a Z score of negative 2. Now what we want to know is what's the probability of getting a Z score of negative 2 or less. So if we look that up using Z tables, we can look them up on the internet. You usually find them in the back of statistics textbooks, funnily enough. The probability of a Z score less than or equal to negative 2 is 0.023 if we were to grab a Z table and look that up. Okay, so what that means is approximately 2 children in 100 would score this low. Okay, so that's an extremely low score. About 98% of the population score higher than that child on that particular test. So it probably suggests they need some assistance. Right. Okay. Looking up where individual scores sit within a distribution has some utility, as we just saw with that example, but not a lot really. I tend to find it most useful when I'm looking at data, when I'm trying to find and identify outliers. Okay. What we often want to do in research, though, is compare groups or group means, perhaps on some experimental intervention. The Z distribution helps with this, as it allows us to use something called the empirical rule to identify the percentage of scores that lie within a certain distance of the mean. Okay, so you can see from this, I got this image from Open Interest Stats book online, and it's kind of really good stats book. I would recommend it. Okay, as you can see from this, you've maybe seen it before, but 68% of scores lie between -1 and +1 standard deviation of the mean on a standard normalized distribution. roughly 95% sit between -2 and +2. It's actually 95% sits between -1.96 and +1.96 standard deviations. So it's probably about 96% of scores sit between negative and +2. So slightly different to what's shown on this particular picture. And there you can see nearly 99.7% of scores sit between -3 and +3 standard deviations of the mean if we're looking at standardized data. So about what we could say though is what's most interesting to us when we're looking at statistical significance is that about 95% of scores lie within 2 standard deviations in the mean. So as I said, actually about 1.96 standard deviations. This is useful for us to know when we're doing research that involves hypothesis testing. Okay. So let's talk about hypothesis testing, or in this case, null hypothesis significance testing. So when we conduct research, we often set up our research so we're testing a hypothesis, such as how effective some intervention, some treatment, some manipulation is that we've given to our participants. Okay. What we do is set up hypotheses to test if the manipulation had an effect. So firstly, we set up a null hypothesis, often referred to as H0, H sub 0 in this case, as you can see on the slide. And this states that the mean for the group, given the manipulation, will be the same as if no manipulation had taken place. So the mean for that group won't be different to the group on whatever variable it is we're measuring, to the group that wasn't given the treatment or manipulation. Okay. We also set up an alternative hypothesis. So H0 is really the status quo. Before I talk about alternative hypothesis, H0 is the status quo. Really, is what we are effectively saying is that if H0 is true, the null hypothesis is true. There is no difference between groups in the population. Whatever manipulation we used had no effect. Okay, and this means that the means between those groups should be roughly about the same. Okay, we also set up an alternative hypothesis, often known as H sub 1 or H sub A, H1 or HA, and this is a statement that the mean score of the group or the individuals given the manipulation is going to be different to those not given it. Okay. In other words, our treatment, our intervention, our manipulation had some sort of effect. When we gave it to them and then measured what happened, their scores were different to people not given this particular treatment. So it could be something like we've got a load of patients with anxiety and we've given some of them a particular psychological therapy or something like that to see if that reduces their anxiety. And then we look, the null hypothesis would be that psychological therapy doesn't work and their anxiety levels will be the same as everyone else. And the alternative hypothesis would be their anxiety levels, the therapy works, their anxiety levels are lower because they were subject to that particular manipulation. To give it a bit of a concrete example. So when hypothesis testing, the question we are trying to answer is when should we accept the alternative hypothesis? We assume that the null hypothesis is true. Okay, if it is true, what is the probability of getting the means for our groups that we got? What is the probability of getting those results? If the probability of getting those results is small, then we can reject the null hypothesis as it probably isn't true. We can believe that the alternative hypothesis H1, H0 is probably true or possibly true. We never really know if H0, if the null hypothesis is false. All we do is really gather evidence against it. Okay, so we want to work out the probability of getting our results. To do this, we use the p-value from our inferential statistical test. Whatever test we use to compare these means, we look at the p-value for that. And the p-value is correctly defined as the probability of obtaining that value of the test statistic, whatever it was, or a value more extreme given that the null hypothesis is assumed to be true. Okay, so the p-value is an inverse measure of the strength of evidence against the null hypothesis. The smaller the p-value, the more unlikely it was that we get our results, and the more likely that the null hypothesis is not true, that we could reject the null hypothesis. Okay. Right. Statistical significance, not much more to cover. So, how low does the probability have to be for us to say it is not due to chance? In many scientific disciplines, the threshold for rejecting the null hypothesis is usually set at p is equal to 0.05. Okay, so that's about a 5% chance of getting those results, or a 1 in 20 chance. Okay, so you should note that this is just a convention, it's not a magic value. The threshold can be adjusted depending on how stringent you want to be. And some disciplines you probably want to be quite stringent. So if it's engineering and there's a risk that whatever it is you're testing might blow up and kill people, you might want to have a more stringent p-value than that. Same in medicine, if you're testing drugs or something like that. You might want to be more stringent before you assess how things affect people. Okay, so a common term that's used to refer to this threshold for the p-value 0.05 is usually p is less than or greater than alpha, and if p is less than or equal to alpha, we can say the result is statistically significant. So the alpha value, the alpha criterion in psychology is this cutoff point. There's a convention that's used of p is less than or equal to 0.05. Okay, right. So some final notes. I'm gonna stop there because there's a lot of material to cover. Some final notes on this particular material. So there are other distributions than the normal distribution. Okay, it has been quite central to a lot of statistics, and it's central, as I said, to a lot of the parametric tests, the most common tests that are used in statistics. But some variables are not normally distributed. Okay, raw data and sample data rarely are normally distributed. Okay, but the sampling distribution of sample means, what's called the standard error, is normally distributed given large enough samples and a population that doesn't deviate too much from being normal. Okay, and errors, or what we call residuals or deviations from the mean, are often normally distributed as well. Okay, and the standard error and what's known as the central limit theorem and confidence intervals and things like that, those are things I'm going to cover in next week's lecture. They're the kind of final conceptual things that I'm going to cover before we can then really get on with learning about specific statistical, inferential statistical tests and applying them to data. Okay, so last week and the first week and this week was lots of conceptual stuff, foundational conceptual stuff, which kind of is a real can of worms once you get into it. It's quite lots of misconceptions, quite easy to get it wrong, essentially. If the last point I've made on this slide here is that if our data doesn't deviate too much from normality, parametric tests are kind of powerful tools we can use to analyze that data and draw inferences. And there are various tests we can use to see how much it deviates. Okay, those are the sorts of things we're going to be doing in the lab classes. Right, so some references, a couple of references for people to look at if they want to. The first one's the Andy Fields statistics textbook. Because we're using, on this course at the moment, we're using SPSS, so discovering stats using IBM SPSS, that's kind of a useful textbook, chapters 1 and 2. The other one that I like that I've put on there, you can access electronic copy with through libraries, the Tim Erdem book, Statistics in Plain English. If you look at chapters 2, 3, 4, 5, 6, it kind of goes over a lot of this stuff. Quite nice explanations. And I had to go through a lot of kind of different sources, resources to sort of piece together what I felt was the best way to sort of explain this stuff. Because, like I said, no matter where you start, there's always probably some prior knowledge that you could have covered, and it's a case of where do you go back and then cover that. Okay, so right, thanks very much. That's it. Hopefully, what I'll do is, in addition to the lecture, I'm going to cover this material in the lecture, but I'll make this audio version available as well as supplementary material, so people can review it if they wish to. Okay.